Okay, today we're going to be talking about the structure and function of large biological molecules, also known as macromolecules. So, of course, all living things are made up of molecules, and in particular, these are large molecules, so they're macromolecules. And the four major groups are carbohydrates, lipids, proteins, and nucleic acids. So we're going to talk a little bit about each of these groups today. And then the lab we're going to do after is a way to help detect these different kinds of macromolecules. So within the cells, there are smaller molecules that are joined together to form larger molecules. So the macromolecules are made up of subunits, smaller pieces that are put together. And once again, we're going to see the importance of molecular structure and function. So something that I mentioned last time, and something you need to remember, is that structure and function go hand in hand, and it's all the different chemical bonds which allow for the structure, right, to form, and then that allows the function. So when we join smaller subunits together, we call those smaller subunits monomers, and we join them together to make a polymer. So the typical reaction where we combine a monomer with a short polymer, also called an oligomer, which just means a short polymer, is a dehydration reaction. So water gets lost when the bond is formed. If we break that larger molecule into smaller pieces, we use water to do that. So we add water, and we call that a hydrolysis reaction. So cells constantly build macromolecules by joining monomers together to form polymers, and cells also break down those macromolecules into monomers, into the smaller pieces. So dehydration reactions are used to join molecules together and release water. And then water can be used to hydrolyze, to break the chemical bonds and separate monomers from a larger macromolecule. So remember, cells have an aqueous environment, a watery environment. So there's a lot of water inside the cell, there's water outside the cell. So water participates in these biochemical reactions. The carbohydrates are energy sources, and we're going to learn a lot about aerobic cell respiration and some other forms of respiration later on. But carbohydrates are the primary fuel source, or the primary food source. So cells can build carbohydrates, and they can also break them down. The simplest carbohydrates are the monosaccharides. Mono means one, saccharide means sugar. So these are the single sugars, the simple sugars. They're just one component. Those monosaccharides can be joined together to form larger saccharides. If you combine two monosaccharides together, you get a disaccharide. If you combine a lot of monosaccharides together, you get a polysaccharide. Now, there is some chemistry here, and I wrote some of the stuff already, although when it gets uploaded into Blackboard, it doesn't make a perfect copy because my handwriting was much neater than what you see here. It's just that in the conversion in Blackboard, it messes stuff up a bit. But when you have a C double bond O, we call that a carbonyl group. And that's something that we're going to see that's very important. If you have a C double bond O and an OH group, that's a carboxyl group. And I did write this properly, it's just, it didn't convert, so it looks messy here. But that is a carboxyl group. So if you go on to take a lot more chemistry, especially organic chemistry, you'll learn a lot more about this stuff. But we have to talk a little bit about organic chemistry since we're talking about these large macromolecules. So sugars are two major types, aldoses and ketoses. So whenever you see the suffix ose, O-S-E, that indicates a sugar. Not to be confused with ASE or ACE, which is an enzyme. So oses are sugars, aces are enzymes. So an aldose and a ketose. So aldoses have that carbonyl group, but it's an aldehyde group. And an aldehyde has a carbonyl group bonded to a hydrogen, and then R refers to anything other than a hydrogen. So any other group. It could be something with carbon, it could be nitrogen, it could be all different kinds of things, but it's not hydrogen. So again, the conversion is messy because that's what Blackboard did to my writing. So an aldehyde has a carbonyl group, it's bonded to a hydrogen, and then an R group, so something else. A ketone also has a carbonyl group, but the carbon is bonded to other R groups, so no hydrogen, it's something else. And so we can see here, our carbonyl group is bonded to two other carbons, and here the carbonyl group is bonded to a carbon, right? That's the R group, and then a hydrogen. So aldoses have an aldehyde group, and a ketose has a ketone group. A triose has 3 carbons, right? Tri means 3. So a 3-carbon sugar, like glyceraldehyde, and we'll see this in the future. We're not going to worry about dihydroxyacetone. Pentoses, penta means 5. So there are 5 carbons, and ribose is an important sugar, and we will see this again. Ribose is the sugar in RNA. Deoxyribose is the sugar in DNA. Ribulose is something we'll see much later when we talk about photosynthesis and the Calvin cycle and how plants make sugar. And then we'll see the ribulose bisphosphate, so that's where that sugar comes in. The hexoses have 6 carbons. And we have 3 hexoses, glucose, galactose, and fructose. They're all hexoses. They all have the same chemical formula. And this is a chemical formula you have to memorize. C6H12O6. So that is the chemical formula for all the monosaccharides, for all the hexoses. So all 3 of these sugars, like glucose, galactose, and fructose, are all monosaccharides, right? They're all monosaccharides, and they are the 3 monosaccharides. They have the same chemical formula, but not the exact same chemical structure. You do not have to memorize the chemical structures. But the hexoses are the monosaccharides. They're all very important. Aldoses have an aldehyde group, and ketoses have a ketone group. But that carbonyl group is something we're going to test for in lab. So remember the carbonyl group. It doesn't really matter whether it's an aldehyde group or a ketone group. As long as that carbonyl is there, we can test for that. That will help us to determine what kind of sugar we have. But C6H12O6 is the same chemical formula for glucose, galactose, and fructose. Although normally when you write C6H12O6, we're talking about glucose. But it could be, the other 2 monosaccharides, but most of the time when you see that formula, it's referring to glucose. So the linear structure can bond to itself. We can see here where we have our carbonyl group, right, the carbon here, and it can loop around and form a chemical bond here, which creates the ring structure of the sugar. So the monosaccharides can exist as either a linear structure or a ring structure. Most of the time, these sugars exist as a ring structure. Now, when it comes to equilibrium, I know, Christopher, I think you asked me a question yesterday. I don't remember what answer I gave, but when a reaction is in equilibrium, that doesn't necessarily mean there are equal amounts on both sides of the equation. So that doesn't mean there are equal amounts of products and reactants. What equilibrium means is that the concentrations are stable, right? They're consistent, they stay the same. In the case of the monosaccharides, the predominant form is the ring structure. More than 99% of the monosaccharides will be in a ring form. Less than 1% will be in the linear form. So in this case, equilibrium means 99% ring structure, 1% linear structure. And that will be stable, that will be consistent. So that, that chemical reaction where they're jumping back and forth between the ring structure and the linear structure, equilibrium is almost all ring structure, just a little bit of linear structure. But they do jump back and forth. But it's mostly going to be the ring structure in solution. But those rings will open up. And when they open up, we can test for that, because when the ring structure opens up, it exposes the carbonyl group. And we'll see in the lab exactly how we can test for that. Any questions on this so far?
So they're more stable when they're in ring form?
Right. It's just the most, it's more thermodynamically favorable. They will kind of bend around and form that chemical bond, but it can be broken relatively easily. Like I said, they do jump back between the linear and the ring forms, just that mostly they're in the ring form. A disaccharide is formed by a dehydration reaction that combines two monosaccharides together. We call that particular type of bond a glycosidic bond or a glycosidic linkage. So it is a covalent bond, but it's between two sugars, so we call it a glycosidic bond or glycosidic linkage. So if you combine two glucoses together, you get maltose. If you combine glucose and fructose together, you get sucrose. And if you combine glucose and galactose together, you get the disaccharide lactose. So the three disaccharides are maltose, sucrose, and lactose. The three monosaccharides are glucose, fructose, and galactose. So you need to know the three monosaccharides and the three disaccharides. Now, polysaccharides, there are three main polysaccharides: starch, glycogen, and cellulose. They are all made of glucose. So starch, long chains of glucose. Technically, they call starch amylose, and that will make more sense because the enzyme that breaks down starch is amylase. So amylose is what we call starch. And then glycogen is also made up of glucose, but it tends to be more branched. Animals, including humans, we store glycogen in the muscles and the liver. So it can serve as an immediate source of glucose, which we need for energy. So if your muscles have a lot of glycogen, you can do a lot more work. If your muscles are deficient or depleted in glycogen, your muscles will fatigue very quickly. It's hard to do much work. So starch we find in plants like potatoes, rice, stuff like that. But animals store their own form of starch called glycogen. Animals have no problem breaking down starch and glycogen. We have the enzymes like amylase to break down starch. Cellulose presents a problem though. Cellulose, as you know, is the component of plant cell walls. Cellulose is just made of glucose. But if you were to eat nothing but lettuce, you would starve to death. Because even though there is tremendous potential energy, because cellulose is all made of glucose, most animals cannot get to it. And that's because we cannot break the bonds between the glucoses and cellulose. So look at the difference here. So glucose, and if you've ever seen the a ball and stick models in chemistry, has anyone ever played with ball and stick models in chemistry? You know what I'm talking about with the balls and sticks, with the atoms and the chemical bonds between them? So the thing about a covalent bond, it's not a rigid structure. Atoms can rotate around the bond. And so if you build a molecule, you can see how you could twist, the molecule around, right? Because the chemical bonds are not rigid, right? They're hard to break, but the atoms will rotate around those chemical bonds. And so glucose can actually exist in two forms. And we call these two forms isomers. So exact same chemical formula. It is the same substance. It just has two different three-dimensional conformations. And so one of them is the alpha form, right, the alpha isomer, and the other one is the beta isomer. So alpha glucose is the form that's found in starch and glycogen. Beta glucose is the form that's found in cellulose. When you join alpha glucoses together, you get what's called an alpha 1,4 linkage. When you join the beta glucoses together, you get what's called a beta 1,4 linkage. Notice that in this molecule here, all the OHs are on the bottom, but in cellulose, they alternate. So the enzyme amylase, it can easily wrap around and cut the bonds between the glucoses in starch and glycogen. It cannot wrap around and break the bonds in cellulose because the OH groups, the hydroxyl groups, are in the way. And so the amylase cannot recognize the cellulose to break it apart and release that glucose. In order to break down cellulose, you need the enzyme cellulase. Animals do not make cellulase, which is why we can't break apart the glucose and use it for energy. So you can eat all the lettuce you want and you'll starve to death. And so you should be thinking, well, cows eat grass and plants, horses eat grass and plants, hay and stuff. Termites eat wood. They're eating cellulose. Look how big a horse or a cow gets. But they don't have cellulase. So how can they possibly get energy from cellulose? Right? Those animals that eat nothing but plants, they don't produce any cellulase. So then how can they survive and actually grow really big eating plants, eating cellulose? Any ideas?
Are their bodies just better equipped to break it down?
Well, but their body, right, they can't, right, because they don't produce any cellulase, right? The animals do not have a gene, for cellulase, and yet they live on nothing but plant matter. And they get a lot of energy from it. Yes, something in their gut biome. So there are many bacteria and protozoa which produce cellulase. And so those animals have these gut microorganisms that produce cellulase and break down the cellulose into simpler sugars. Then those animals can absorb it. So the only reason why those herbivores are able to survive on plants is because they've been colonized by microorganisms in their gut. And it's the microorganisms that digest the cellulose for those animals. Humans do not have any of those microorganisms, so we cannot break down cellulose, right? Now, when you think of beavers, the beavers that, chew down trees and they build dams and stuff, and, they eat bark, they eat wood. So here's a little bit of a problem. The beavers do have those microorganisms in their gut, but they're at the end of the gut, in the colon, in the large intestine. And they can't absorb any sugars from the large intestine. But that's where the gut microorganisms are that break down the cellulose. So how does that work? If the gut microbes are at the very end and they're breaking down the cellulose, but it cannot be absorbed, through the colon, then how can beavers possibly get any energy from the wood, from the bark that they eat? They have to do something. What do they have to do?
They eat their poop.
Right, they eat their poop, right? So after all the bark passes all the way through the intestine and the microorganisms have had a chance to break it down, the beavers poop it out and then they eat the poop, right? Because now it's been processed, right? The cellulose, a lot of it has been broken down. And so when they eat that, now they're able to absorb the simpler sugars. So nature finds a way to accomplish everything. Waste not, want not, right? But the key point here is that cellulose has a form of glucose, the beta form, which forms a different glycosidic bond. It forms a beta 1,4 linkage, not an alpha 1,4. As a result, animals do not have the enzyme, right, which can break down the beta 1,4 linkage. So that's why cellulose is not a good energy source for humans, right? Because we can't break it down. And when it comes to animals, the animals themselves do not break down the cellulose. It's the gut microorganisms that do it for them. And that's the only reason why all those herbivores can eat nothing but plant matter and grow pretty large too. So again, cellulose is a structural component of plant cell walls. So there's tremendous potential energy in plants. It's just that we can't get to it. So when it comes to agriculture, if you think of something like corn, right? We've all seen fields of corn, right? Those tall, 6-foot tall corn plants, right? All those big plants. We've all seen corn plants, right? Fields of corn. So that big, huge plant, how many ears of corn are on that big, huge plant? We're talking about the field corn you've seen growing by the road. So how many ears of corn? No, not 5. It's usually just 1, sometimes 2. But that big, huge corn plant, it normally doesn't produce but 1 good ear of corn. So what do you do with all the rest of the plant? Now, other types of corn selectively bred by humans for humans to eat, and then the corn plants produce more than 1 ear. But the ordinary field corn, the original corn, only produces 1 ear per stalk, usually. So, and that's what you see growing in the fields, is just field corn. So you got that big, huge plant. What do you do with all the rest of that plant matter? Just throw it away, bury it, you compost it? You can compost it, right? Well, what scientists have done, you have food for animals, right? Pigs will eat anything, but they can grind it up and use it to make food for animals, or they can use bacteria to produce cellulase, and they can break down that excess plant matter into simpler sugars and then ferment those sugars to make alcohol, which can then be used to make ethanol fuel for cars. So this excess plant matter, a lot of it is being used, to make ethanol fuel, because we can get the enzyme from bacteria and mix it with all this useless plant matter and break it down into simpler sugars and then ferment it. So nature provides what we need. We just have to learn how to use it. So cellulose has tremendous potential energy in it with all that glucose, but animals cannot get to it directly. We need gut microorganisms to help break down the cellulose into simpler sugars. But our 3 polysaccharides are starch, glycogen, and cellulose. Animals can break down starch and glycogen, but not cellulose, unless you have microorganisms living in your gut, and then you can break it down. Now, insects and fungi insects have an exoskeleton, the outer skeleton, and fungi have cell walls, but they're made of chitin. And so chitin does involve a sugar and an amine. And so we call this N-acetylglucosamine. And so this is what chitin is made out of. Now, N-acetylglucosamine is also a structural component of many bacterial cell walls. Peptidoglycan in bacteria includes N-acetylglucosamine. So that was there, and the bacteria used it in their cell walls. Insects use it in their exoskeletons. Fungi use it in their cell walls. And so again, the fact that we have all these different organisms using the same kind of structural component would suggest a common ancestor, somewhere in the distant past. And so this molecule was used to make peptidoglycan in bacteria, or to make chitin in insects and fungi. Chitin is strong and flexible and can be used as a surgical thread, right? So we can use it to sew up, wounds in people. And insects are very nutritious, right? So you can eat them, right? They're crunchy, right? The chitin. Some of you like to eat nasty mushrooms, right? Which have a lot of chitin in it. So it's edible and digestible. So those were carbohydrates. Moving on to lipids, these are molecules that are hydrophobic. We talked about that last time. Water hating. So these things do not like water. They don't like to act with water. So lipids are large biological molecules, but they don't really form polymers. although there are some subunits you put together to make them, but they're not really polymers. So they don't like water, they don't mix with water, right? They have nonpolar covalent bonds, so there are no charges or partial charges which can interact with the water. But when it comes to biological lipids, we have fats and oils, phospholipids, and steroids or sterols. And so those are the four major groups that are important. So here we see fats. So a fat is composed of a molecule of glycerol and at least one fatty acid. One fatty acid, and we call this a monoglyceride. If there's two fatty acids, it's a diglyceride. And if there are three fatty acids, it's a triglyceride. Technically, it's a triacylglycerol molecule, but we just call it a triglyceride. So a molecule of glycerol and at least one fatty acid makes a glyceride. So one fatty acid, monoglyceride, two fatty acids, diglycerides, and three fatty acids are triglycerides. We're going to focus on the triglycerides because we eat them and we also store them as fat in our body. So we store triglycerides. Notice the long hydrocarbon chains in the fatty acids. So these are hydrophobic, right? They don't like water. You probably have all heard of saturated and unsaturated fatty acids or fats. The saturated fats are the ones that are bad for you, that cause heart disease. And then the unsaturated fatty acids are the ones that are good for you, the good fats. So the word saturation refers to hydrogen. So a saturated fat means that all the carbons are bonded to as many hydrogens as possible. So we see here, and we saw it before here, that all the carbons are bonded to hydrogens here. So this is a saturated fat. It's saturated with hydrogen. Saturated fats, all those fatty acids are linear molecules, which means that the triglycerides can all stack together and pack themselves very tightly together. And that's why saturated fats tend to be solid at room temperature, because all these long linear, hydrocarbons are able to pack tightly together. An unsaturated fatty acid has at least one double bond between two of the carbons. So because there's a double bond, that means there's two fewer hydrogens that are able to bond. So fully saturated, we say it's unsaturated. So one double bond, we have a mono unsaturated fatty acid. If you have multiple double bonds, you have a poly unsaturated fatty acid. When you put a double bond into the hydrocarbon chain, it creates a kink, a bend. That bent molecule means that the fatty acids cannot pack closely together. They cannot pack tightly together. They stay further apart. And that's why unsaturated fats tend to be liquid at room temperature. So they're oils. So that means they're unsaturated. Right? So saturated fats like butter, solid at room temperature, because all the carbons are full of hydrogen. All the molecules are nice and linear, so they pack tightly together. But unsaturated fats have these kinks. They have the bends in the fatty acids, which means that the fatty acids cannot tightly pack together. They can't get as close together. And so the fat is unsaturated it's a liquid at room temperature. So those are the oils. And those are healthier for you. Now, you can take an unsaturated fat and artificially saturate it. And we call those trans fats. So even though they're unsaturated, they've been artificially saturated. And trans fats are also very bad for you. And so I think here in the US, we pretty much eliminated trans fats from almost everything, so we don't use them anymore. But in some countries, they still do. So you can take a healthy, unsaturated fat like oil, and you can artificially saturate it and make it solid. And that's what we call a trans fat. And that's an unhealthy fat as well. A phospholipid, these are the lipids that make up membranes. So a phospholipid has two fatty acid tails. It has a molecule of glycerol, but it has a phosphate group and a choline group. Notice that the phosphate and choline are charged. So because they have charges, they like water. They like to interact with water. So the head of a phospholipid is hydrophilic. It loves water. But the tails are still hydrophobic. They do not like water. So if you take some phospholipids and you mix them in a beaker of water, they always form a characteristic structure. And that structure is a bilayer. So here we see the lipid bilayer. So this bilayer configuration is what always happens when you mix phospholipids with water. The hydrophilic heads, they face outside to interact with water, and the hydrophobic tails interact with each other because they hate water. So the tails, face each other to minimize contact with the water. And so this is why we have lipid membranes. They're made of phospholipids, right? The inside of the cell is watery, it's aqueous, and the outside of the cell is watery and it's aqueous. And so that's why cell membranes are made of phospholipids. If you take a bunch of phospholipids and vigorously mix them with water, you'll create some little bubbles where some water is completely surrounded by the phospholipid bilayer. So think about that. You got a bubble of water surrounded by lipid membrane. That's similar to what a cell is, right? And so that's one of the ways they think that cells evolved, is that as the phospholipids evolved, they partitioned the inside from the outside, right? So the inside was separated from the external environment, and that helped to lead to the formation of cells. But the lipid bilayer configuration is a function of a hydrophobic tail and a hydrophilic head on that phospholipid molecule. And we're going to learn about membranes next time. Now, sterols or steroids are also extremely important. And the most common sterol is cholesterol. We've all heard of cholesterol, right? Is cholesterol good or bad?
You don't
They're
want
bad.
Well, cholesterol is both good and bad. It's good because it's essential. We need cholesterol, right? Cholesterol helps maintain membrane integrity. Cholesterol is also converted into the sex hormones, testosterone, and one of the estrogens. So they're based on cholesterol. So we need those in order to make the steroid hormones testosterone and one of the estrogens. Vitamin D is also synthesized from cholesterol. As you may know, we make vitamin D when our skin is exposed to ultraviolet radiation. There is a reaction that occurs where cholesterol is converted into vitamin D, which is an essential vitamin. So without cholesterol, we have no testosterone, right? We're missing one of the estrogens, and we don't have any vitamin D. So cholesterol is essential. The problem is we make all the cholesterol we need. When you eat excess cholesterol, that's what leads to heart disease and other problems. So consuming a lot of excess cholesterol is bad. So a lot of red meats, fatty meats, eggs, as long as you don't eat more than 1 or 2 eggs a day, that's not a problem. But if you eat 6 or 8 eggs a day, whole eggs, that's a lot of extra cholesterol. So that would not be a good thing. But 1 or 2 eggs a day is not a problem. It doesn't affect your overall serum cholesterol. But normally, most people make all the cholesterol they need. So excess cholesterol is a problem. It can lead to heart disease. Now, some people have a condition where they produce too much cholesterol. So some people naturally produce way more cholesterol than they need. So they are naturally at higher risk for heart disease. And you can get tested for cholesterol, and there are medications to lower your cholesterol if yours is a problem. But most of you are young people, so you probably never had your cholesterol tested. But it should, be below 200, your total cholesterol, and the further below 200, the better. But you guys are young, so you don't have to worry about that yet. But as you get older and you have blood work done, they'll test you for cholesterol to see if it's okay. If it's too high, they can recommend dietary changes, exercise. If it's way high, then they'll give you medication to bring it down, right, to reduce your risk of heart disease. But cholesterol is essential. We need it. It's just that we normally make all the cholesterol we need. So excess cholesterol is the problem, not just cholesterol. When it comes to proteins, proteins serve a multitude of functions inside of cells. All enzymes are proteins. We have many structural proteins in our cells, in our bodies. Our collagen, our muscles, right, are made of protein. Storage proteins, transport proteins for carrying substances around the body or moving things into and out of cells. Hormones, like insulin, is a protein. We have all kinds of receptors on the cells, which are proteins. I mentioned muscles. And we also have defensive proteins, the antibodies, which protect us against all kinds of things like COVID, right? So those are proteins. So proteins serve a variety of functions inside of cells. So they're very important. So a fully formed string of amino acids is a protein, but as the protein is being synthesized, we call it a polypeptide, and then after it's modified and fully formed, then we typically refer to it as a protein. So a polypeptide you can kind of think of as an incomplete protein. It's not ready to go yet. When it's all ready and fully functional, then we call it a protein, but sometimes we use the words interchangeably. But the polypeptide or proteins, they consist of amino acids, but there are 20 amino acids that make up proteins. There are a couple more naturally occurring amino acids, but they're not involved in protein synthesis. They're involved in other reactions inside the cell. So the amino acids that make up proteins, there's only 20 of them. And then, of course, humans have created some synthetic amino acids, but there are 20 naturally occurring amino acids that are used to make proteins. So an amino acid has an amino group and a carboxyl group. In between is a carbon, which is bonded to a hydrogen and then another R group, right? So it's bonded to something else. But this is the overall structure for amino acids. Now, when it comes to discussing amino acids, it's only considered an amino acid when it has the amino group and a complete carboxyl group. When you join the amino acids together, right, to make a peptide or polypeptide, technically, it's no longer an amino acid. And so what do they call these things when they're actually bonded together? They call them residues. So an amino acid residue. I bring this up now because we're going to see that later on when we do bioinformatics and we're looking up the protein structures and the amino acids. And they don't use the term amino acids because it's no longer an acid. When you combine it to other amino acids, it's no longer an acid, so they call it a residue. Right, so just FYI, we'll see residue again. So here we have all the 20 amino acids. So if you're a science major and you go on to take biochemistry, they typically make you memorize the structure of all 20 amino acids. So imagine having to memorize all of these 20 different chemical structures. I had to do that when I took biochemistry. I don't remember them now, but I did have all the structures memorized at one time. So now, all these different amino acids are different because of the R groups. And so we have non-polar amino acids, we have polar amino acids, and we have fully charged amino acids. Some are positively charged and some are negatively charged. So all these different amino acids have different chemical properties. And that's what causes the different polypeptides to fold in different ways to make different proteins. And it's also all these different kinds of amino acids that allow proteins to perform their functions, right? So remember, structure and function and the chemical bonds. When we talk about enzymes, the active site of an enzyme has certain amino acids in it which can interact with a substrate. And the interactions are all those different kinds of chemical bonds, mostly hydrogen bonds, but also ionic bonds. But bonds form in the active site. When it comes to receptors, right, those protein receptors, which bind to hormones or other chemicals like morphine and heroin, whatever, it's because of the chemical bonds that occur between the amino acids in the receptor and whatever the molecule is that's binding to that receptor. So all these different amino acids have different chemical properties. That's what allows proteins to fold in the characteristic ways that they do and allow them to perform their functions. So in order to make a polypeptide, you have to join together amino acids. We do that with a dehydration reaction. And the bond that's formed we call a peptide bond. So when you join the amino acids together, we call them a peptide bond. And I said, they're no longer considered acids, they're now called residues. we can test for the presence of peptide bonds, to determine if something has protein in it. And we'll see how to do that in lab today. So when you join two amino acids together, you form a peptide bond. And so that's how we get a polypeptide. So again, structure and function. So here we're looking at an antibody, and we're looking at a model of it. And antibodies are very specific to a particular pathogen, or more generally an antigen. And the reason why they're so specific is because there are antigen binding sites on the antibodies. And those binding sites form chemical bonds with the antigen. And so that's why the antibodies stick to a specific antigen. So antibodies are very specific, right? They don't just bind to anything. One antibody binds to one particular antigen. And since there are, countless millions of potential antigens out there, we all have the ability to produce millions of unique antigens, each one specific for one type. We can produce millions of unique antibodies, each one specific to one type of antigen. And that's because of the chemical bonding that occurs between the antibody and the antigen. So structure and function, and the chemical bonding that occurs. There are 4 levels of protein structure. The sequence of amino acids, we just call that the primary structure. It's just a sequence of amino acids. This sequence can fold on itself and form 2 secondary structures. We have alpha helices and beta sheets, or beta pleated sheets. So these are the 2 secondary structures. And these secondary structures are not just random, they actually serve a purpose. And again, much later on in lab, we'll see how the alpha helices and beta sheets can stabilize proteins, make them either more rigid or more flexible, depending on the percentages of alpha helices and beta sheets. So they do serve a purpose. When everything folds on itself to form a 3-dimensional globular structure, we call that the tertiary structure. And for many proteins, that's it. They're done. They're finished. They're functional. But there are some other proteins that require more than one subunit to associate. If that's the case, we call that the quaternary level of protein structure. So the primary level is just the sequence of amino acids. There are 2 secondary structures, the alpha helices and beta sheets. When everything folds on itself, we call that the tertiary structure. Many proteins are finished at that point, but some other proteins require more than one subunit to associate together, and that forms the final functional protein, and that's the quaternary level of protein structure. So if you can destroy the quaternary, tertiary, and secondary levels of protein structure, you have denatured that protein. You have destroyed it, right? Because most of the time, once you cause that protein to unfold, it normally cannot refold by itself and go back to its normal shape. So we'll talk about how we denature proteins later, but heat is a good way to denature proteins, right? You cook something, you denature it. You're actually breaking the proteins apart, and so they cannot renature. We can use acids and bases, right, to destroy protein structure. If an environment becomes too acidic or too basic, you'll break apart the tertiary structures, right, the secondary structures. You'll denature those proteins and destroy them. So all the different kinds of amino acids associate to form different types of chemical bonds. We'll have hydrogen bonds, we'll have ionic bonds, we'll have some covalent bonds, like between two sulfurs. There are some sulfur-containing amino acids which create what's called a disulfide bridge. There's also van der Waals forces, right? So all those different kinds of chemical bonds and interactions cause a peptide to fold in a very specific way. And it's that folding that gives that protein its structure and its function. That's all because of all the different kinds of amino acids and all the different chemical interactions they have with each other. Hydrogen bonds, covalent bonds, ionic bonds, van der Waals, right? So they all play an important role in structure and function. So remember, chemical bonding is what creates the structure, and that structure can perform a function because of chemical bonding. So, as I mentioned, we can denature proteins, usually with high heat or acid or base. Most of the time, once you've done that, you have destroyed that protein. It cannot renature. Certain proteins can renature, or there are other proteins inside of cells that can help a large protein to refold again, to become functional. But normally, once you denature, you have destroyed. An example of how certain cells can survive the heat, certain bacteria, if you heat them up to the point where it's extremely unpleasant for them, not to completely boil them away, but you heat them up enough to the point where it does a lot of damage, the bacteria start producing what are called heat shock proteins, which help to repair the damage inside the cell. So there are cellular mechanisms that can help certain denature proteins to renature and refold and become functional again. But when you fry an egg, you can't put that egg back in the shell. You've completely denatured it. You're not going to ever be able to renature that egg. It'll never be the way it was inside that shell after you fry it. It's completely denatured. So here we're just looking at an image that's summarizing how DNA is used to store genetic information. And as we'll learn later on in the course, the DNA gets transcribed into RNA, which is then translated into protein. And so we're going to see several macromolecules in action, right? Because we have the DNA, RNA, a lot of proteins and enzymes are involved in transcription. Ribosomes are involved in translation to make those polypeptides. And so we'll see how all that comes together later. But nucleic acids, DNA and RNA, are extremely important. They store all the genetic information in a cell that's needed to create a new organism. And so nucleic acids are made up of nucleotides. A nucleotide has a phosphate group, a pentose sugar, so it's got ribose for RNA and deoxyribose for DNA, and then a nitrogenous base, our ATGC, and in the case of RNA, U. So we have our pyrimidines, cytosine, thymine, and uracil. In RNA, there's no thymine. It's replaced with uracil. And two purines, adenine and guanine. So these are the nitrogenous bases, and they combine with deoxyribose and phosphate to make DNA, or ribose and a phosphate to make RNA. And we'll learn a lot more detail about DNA and RNA later. Any question? So the DNA molecule is made up of two strands of nucleotides, and the nucleotides are held together with covalent bonds, but the two strands are held together with hydrogen bonds. And so there are hydrogen bonds occurring between the two strands which hold them together. But hydrogen bonds are not very strong. There is strength in numbers, but the idea here is that the cell needs to be able to pull the two strands apart for DNA replication as well as transcription. So the hydrogen bonds are strong enough to keep DNA together, but not so strong that the cell cannot pull the DNA apart so that it can replicate it or transcribe it. And we'll learn about replication and transcription later. So DNA stores all the genetic information, right, to create a new organism, and DNA is passed from parents to offspring. And we're going to learn a lot about genetics later in the course. And in the lab, we talk about the bioinformatics. We'll talk more about how we can determine relatedness in organisms by comparing, DNA sequences or amino acid sequences, right? Because the more similar the sequences are, then the more closely related those two organisms most likely are. So to summarize, all the four major biomolecules or macromolecules are the carbohydrates, lipids, proteins, and nucleic acids. All of them, except for the lipids, are polymers, right? They're made of multiple repeating subunits, right? Carbohydrates are made of monosaccharides, right? The disaccharides and polysaccharides are made of monosaccharides. Proteins are made of amino acids. DNA and RNA are made of nucleic acids. Lipids are not really made up of subunits. When it comes to triglycerides, you do have mono, di, and triglycerides, 1 fatty acid, 2 fatty acids, and 3 fatty acids. And they serve a variety of functions inside the cell. And the cell has to make all these different macromolecules. It does so with dehydration reactions, right, to combine smaller molecules to make larger molecules. And cells will also break down these larger molecules, right? The cells will break down starch into glucose, right? Cells will break down proteins into amino acids, and cells will also break down nucleic acids into nucleotides. And so water is used to split all those molecules, and we call those hydrolysis reactions. So sugars, all those monosaccharides are bonded together with a glycosidic bond or glycosidic linkage. Lipids of the fatty acids are joined to the glycerol with an ester bond or an ester linkage. And proteins, all the amino acids, are joined together with peptide bonds. And in nucleic acids, it's the phosphate groups that join together with the next nucleotide to create a phosphodiester bond. So phospho because the phosphates are being used to help make those bonds. So all these different types of bonds are covalent bonds, right? So they're just sharing electrons, like any other covalent bond. We just give them the special names to let us know that a glycosidic bond means two sugars, right? Esters are involved in all different kinds of things, but here, for us, think of lipids, peptide bonds in proteins, and phosphodiester bonds in nucleic acids, DNA and RNA. So what is a polymer and what is a monomer? So somebody tell me.
is sort of like a string of monomers.
Yep. So the monomers are the subunits which are joined together to form a polymer. So a polymer is a chain of repeating subunits. Monomers are connected in what type of reaction? So how do you join two monomers together?
Hydrolysis.
To join together? Right, a dehydration reaction. Right, that's how we join molecules together, or the monomers together, is with dehydration reactions. Right, so water actually gets formed. And in order to split, right, those large polymers, those large macromolecules, now we use the hydrolysis reaction. So water is used to break those bonds. Right, so dehydration reactions are used to join molecules together, and the hydrolysis reactions are used to break bonds and to reduce those large molecules into smaller pieces. And cells do both. Cells build molecules and cells break them down. So we have 3 disaccharides. What were the 3 disaccharides?
Lactose, sucrose, and maltose.
Right. Lactose, sucrose, and maltose. So lactose is made from which 2 monosaccharides? Yeah, Lauren?
Lactose is formed from glucose and galactose.
Yep, glucose and galactose. What about sucrose?
Is it glucose and fructose?
Yep, and fructose. And maltose?
2 glucose.
Maltose is 2 glucose.
Yep, 2 glucoses. So the 3 monosaccharides are glucose, fructose, and galactose. And what are the 3 polysaccharides?
Starch, glycogen, and cellulose.
Yep, starch, glycogen, and cellulose. And they're all made of glucose, although cellulose is made of beta glucose, that other form. When it comes to the levels of protein structure, the primary structure is just the sequence of amino acids. What do we call this structure here?
Secondary structure.
Which secondary structure is it? Which secondary structure is it? Nope. This is not a helix here.
The beta pleated sheets.
Yep, it's the beta sheet. And what about this one? No, but this structure. And now what do we call this one? Right, the alpha helix. And so these are both our secondary structures. And what about this guy here? It's 3-dimensional, it's globular. What level?
Tertiary.
Yep, it's a tertiary. And notice here we have two subunits associated together. So what do we call that? Right, the quaternary. So the four levels are protein structure. The primary is just the sequence of amino acids. There are two secondary structures, alpha helices and beta sheets. When everything folds on itself, we call that the tertiary level. And for many proteins, that's it. They're done. They're functional. But other proteins require multiple subunits to associate right together. And that's the quaternary level of protein structure. So any questions on the biomolecules or macromolecules? Nope.